forward and backward with this? Okay, so those that know me know that at the lab they call me Dr. Jekyll and Mr. Hyde. And this is because sometimes I'm talking about applications, that's when I'm Dr. Jekyll, and sometimes I'm bombarding people with equations, that's when I'm Mr. Hyde. Today I'm going to be Dr. Jekyll because Thomas asked me to talk about multimodal databases, why they're important, and what's their use in translational research. And I'm going to illustrate, since we have such short time, basically from the work of the Cuban Neuroscience Center. Well, brain mapping has hit the news, and though we all know about this for more than 20 years, we now see these huge brain projects. We see Obama announcing the Brain Project. We there see Henry Markram. He's smiling probably because he got $1 billion for 10 years. And then, basically, they're quite interesting projects. For example, a number of years ago, I showed some of the people from the project, specifically Sean Hill, my analysis of the creation of IT platforms, how they're going to collect mouse and human data, how that's going to feed into cognitive architecture, math and theory. It's really a fascinating, let's say, top-down exercise. I have only one problem. In human data, no EEG. So we go on, MEG, yes. So I think the threshold is that if the system doesn't cost more than $3 million, you don't use it to gather data. Well, the Brain Project had started very bottom-up. We had discussions in Seattle, if you remember. It's sort of gotten a theoretical handle. This is when it first came out. And at least in principle, there's the idea of gathering some electrophysiological data. However, one of the big questions is that both have been posed as impacting on the global burden of disease. And as we'll move forward, we see that I think it's only the EEG that could provide that bridge or that link. And the gauntlet that I'm setting out here, and I'm throwing at people's feet, is that we have to include in the project's electrophysiological recordings. Let me see if I can give you some evidence to support this idea. So basically, this is a list of the recent brain projects, and if you start ticking which one has electrophysiology in terms of EEG, not MEG, I think the only one is the Cuban project that's on that list, at least officially stated. To give you a further idea of database collections, here's a timeline. And one of the things I want to remember people is that as early in '73, Matushak and Patterson already started doing the sort of things that later became part of the brain mapping initiative. That is to say, they gathered data on randomized sets of normal and control and disease subjects. They created regression equations for spectral parameters. You've heard all about that. I won't describe them here. Moreover, they used something that is now known as voxel prediction or statistical learning. They tried to predict the age of the person from his EEG. So that started all out on a smaller scale in Sweden here. Later, we had in Cuba and in brain research labs at NYU, we gathered together a series of data. This was an integrative database. There was a science paper in '77. But all these studies had the characteristic of being carried out when MRIs were not readily available. So that led to a second larger project, which I'll describe, which was the Cuban human brain mapping project that started very early on, but only gathered EEG. You'll see how we got around the problems there. Well, these are the timelines of the other ones, and I have to say that the majority that I've looked up don't gather EEG. We started a second one, which I'll describe, a second Cuban human brain mapping project. We did add the EEG together with the more fancy types of neuroimaging, which I love. I really love to play with that data. But It is one of the few projects. If anybody finds out or can tell me about another one, I'll add it to this. So our first project, it says the second one there, but it was actually the first one in 1989, and basically with a Cuban system that we had designed for gathering EEG, we gathered the EEG, we did topography, and then we went on to refine what Roy John and Matuszek and Patterson had done before, which was for each electrode to derive a developmental surface of the average power. So this is all frequencies, this would be the amplitude of the spectrum, and this would be from 5 to 97 years. And these maps were available for each and one of the electrodes. We use this to then apply something that you guys are very familiar with, which is transforming the data, maybe with a log transform first, to look at a distance, which is essentially the Z transform, which is to take out from the measure the average value for that age, for whatever covariates you want, and divide by the standard deviation. This is an early work by Roy John that shows how you can avoid false positives and false negatives by correcting the Z-score with as a function of an age-dependent and standard age-dependent standard deviation and mean. So this was pretty standard. This was done, as you see, in the 80s, and in fact, let's see here. Oh, I did something wrong here. Okay, so we then went on to do the equivalent of randomization tests. So this is a paper that we published for brain topography, and this was, let me see, about 1994. And what we did was to create, on the basis of that Multimodal distance that's now in all the multivariate methods, projecting it onto the electrodes and getting a map with simultaneous confidence intervals. Now, this stuff was really quite interesting. We created a network of laboratories of clinical neurophysiology all over Cuba with Cuban systems, and we started evaluating the statistical usefulness of this in databases that not only included the 5 to 97-year-olds that were controls, but also different groups. I will be showing the ROC plot information that comes from plotting the probability of a true hit, that's say of a true positive, versus the probability of a false positive. And what I'm going to be doing is looking at the area under this ROC curve. So that summarizes the information. If it's 0.5, the test is worthless, be it univariate or multivariate. The closer to 1 it is, 1 would be perfect. Lower than 0.5, you're actually doing something completely wrong. You're diagnosing in the other direction. But that happens. So here's one of the first results of using this statistical methodology. There's an organization called the International Society for Brain Electrical Tomography, Lopez da Silva, Dietrich, a lot of my friends were there, and I asked them all to sit down and get a randomized presentation of maps. This was at the ISBET meeting in Havana in '93. And here we have, with their standard deviations, the ROC curves. So you see that for tumor, for epilepsy, we're doing pretty good. For alcohol, the algorithm people had in their minds was the opposite of what they should have been using. So this is done very rarely. I know the people in radiology do it. I don't think that the people that are doing electrophysiology do this type of testing sufficiently. So for that reason, here's in the book by Carl Friston, he acknowledges that he picked SPM as the name of his package and the methodology as a nod to the significance probability mapping that had been developed by a previous generation for the EEG. This is something people don't remember very well, but it's quite interesting because many of the methods were similar. Now, today, I think, Riera, explained the EEG inverse problem. We're trying to deal there. And this is where I say that I'm Mr. Hyde, but I'm not going to be today. We look at sources instead of the superficial representations. And that there are many different methods. They differ on the assumptions. So basically repeating what probably Jorge said, I don't know if he said it, if you have one electrode and two generators, and this is the lead field, is the coefficient that reflects sources on the scalp, you can have infinitely many different solutions that will give you the same voltage. So any combination of these with these values of the lead field will give the same voltage value. And you have different flavors of inverse solutions by putting different constraints. We put a combination of constraints there, and what we did was then to move from looking at brain topography to brain tomography. And here we see what we presented at the 1996 conference of biomagnetism, which was localization of the alpha rhythm on a realistic head model with a realistic cortex. It was a cortical model, and it was localized in the visual cortex. We were lucky. If anybody wants to, I'll tell you why. And the reason is that it happened to be very large. We later found out that if you don't do statistics, there's an enormous bias towards that can't be solved by many of the usual methods of deeper sources that can really make a weaker source that's more, let's say, more in proximity with the electrodes just jump out. And one of the ways of avoiding this is statistics, but I don't have time to talk about that. So basically, we were lucky, we got this, and then we decided to unite these two projects. So ICBM had begun gathering the data that led to the Montreal Neurological Institute average brain and then probabilistic atlas, and Keith Worsley and Alan Evans joined forces with us, and what we did was this. We started projecting the sources onto this average brain and getting a model which was not with a truly realistic head model. Now, was this of any use? I think so. And in fact, here we see a paper of the group where we actually obtained the norms. We're looking here, this graph that you saw for the occipital region on the scalp now is for the calcarine sulcus, and you see how alpha grows and then decreases in the occipital area with considerable more sharpness in terms of its localization. We then defined what we called Z-maps, which is just the extension of Z maps on the head. Every single voxel, we subtract the mean, divide by the standard deviation for that voxel, set thresholds, and you can see that this already is the same elements of SPM. Now, we then have been using this in several contexts. I want to present one paper which was published a couple of years ago with Talia Harmony, who was one of the people who created our group and is now in Mexico. And this was looking at the patterns of EEG in sleep for babies from 1 to 2 years. And what I've done here is collapse. So for each frequency, we've stacked the, let's say, a maximal intensity projecting a glass brain with the values of the regression coefficients with age from 1 to 2 years. What did we use? We used the recently developed MNI templates for children in that age. What we found was that for very low frequencies, there was a decrease during the first year, then there's an increase in this band, and here in beta, it goes down again. Now, we can use this to try to detect by the Mahalanobis distance whether a child is or not is, let's say, with an abnormal EEG. And what we found was the following. Okay, these are the kids in the box, and this is the map of the Z image from which we calculated the Mahalanobis distance. Now, the area under the ROC curve, and please look here. This is scalp information. So, I'm sorry, this is scalp information here in the middle. This is the Laplacian. And this is classifying the kids according to the sources we are finding. Now, this was bootstrapped. I can give you the details. It's in the paper. In fact, one of the things we did in the paper was to extend the whole idea of power analysis that's useful for fMRI to EEG tomography, as we call it. And that shows there's an equivalence, even if you use an approximate head model. So with this in our hands, we decided to do a new project more recently. And this new project, which was the second Cuban human brain mapping project that started in 2004, was also a random sample of the Cuban population. We went into the ID registries, came out with a perfectly random sample, and then went and knocked on people's doors. And I have to tell you that one advantage of being in Cuba is that people love to be scanned. So we got 90% compliance without paying anything. Well, many of them, what they did is they wanted their heads, their MRI to put in the bed in the living room. But that was okay. We just printed it out for them and gave it to them. And of course, anybody who passed these different stages who had a problem was dealt with by the Cuban medical system. So the samples dwindled a bit, but we managed to gather blood for genomic analysis, EEG with our 128-channel system, and MRI using a 1.5 sample system, Tesla machine. Now, we studied these populations in the context of several studies of disabilities that have been carried out in Cuba. Not everybody has imaging, but we are doing follow-ups on most of them. And so this was an initial group we randomly sampled. You see we have there cognitive endophenotypes of schizophrenia. We have elderly controls in addition, because that was we also studied all the murderers in Cuba. The government asked us that we study every single person which was highly violent. And I feel proud that one thing we managed with the information we gave of the biological basis is that without having left the legislation of Cuba, the death penalty has not been applied since the end of the study. And I mean, it wasn't just, when I say we, I'm not saying the Cuban Neuroscience Center, I'm saying the research establishment of Cuba. We've studied kids a lot and so forth. Okay, so let me show you some examples of these studies here. One of them, everybody, we have an automatic pipeline. Some parts of the pipeline are from the Montreal Neurological Institute. Some are ours. This is an add-on we put on the SPM for analyzing volumes. This is graph theoretical methods developed in our group for looking at tractography. So for the same parcellation you make of the volumes, you can have a matrix of connectivity, and we have this for the 400 subjects that are in the database. We also obtain head models for each subject, now individual head models, and here we obtain the inverse solution. So now we're doing things, let's say, more properly from the strict point of view, not using an approximate head model. I'll speak of this, when we deviate from this, I'll speak about it as we go on. So, some interesting stuff that comes up by also having the EEG. This was a case from Colombia, the country of Colombia. This guy had in a car accident. Here you see 2 fusiform areas wiped out completely. This was quantified morphometrically. This is a paper in Neuroimaging. But furthermore, when we looked at the fMRI and we looked at the electrophysiological recording, what happens is that this guy had covert recognition of the faces he said that he didn't recognize. So using fMRI-seated tractography, this is one example of the normal controls in the database, and you see that the guy who has prosopagnosia still has, from the occipital face area, the ILF practically preserved, and we think that's what explains this. Now, the interesting thing about this is that having a multimodal database allowed us to then create in silico lesions. So what did we do? We started destroying in the computer lesions in the normal, seeing which tracks would degenerate, and then comparing statistics. It's in the Neuroimage paper. But it shows that the more multimodal the database, the higher powered analysis in individuals you can do. Now, okay, in the study of violence I mentioned, this is a very interesting analysis of the area under the ROC curve in trying to distinguish between people that had psychopathic, let's say, behavior versus those that didn't. And what's interesting here is that if you use the EEG and the Wechsler test, you're getting just as good or slightly better performance if you use the MRI plus everything else. Now, I'm not saying we don't need the MRI. I'm just saying that in each case that we want to do something, we have to look at the information content of the data. And the only way to do it is to have multimodal data. So we're missing out on a lot when we're not recording the EEG. Now, we can answer theoretical questions. So this is one theoretical study we published. I was just a couple of days ago, I saw Paul Nunes, who's a good friend. He has the theory that the alpha rhythm, the peak of the alpha rhythm, how fast, how many hertz you have of alpha rhythm, depends on the size of the cortex. And these are his equations. So he's being Mr. Hyde, not me. Now, the thing is that he says that the larger the surface, the more time it takes for a traveling wave to go around, and therefore the slower the alpha rhythm. Well, now we're in a position to test this. And in fact, this is what we did. We tested this prediction, and what we found that this equation, which says that alpha should be slower with a larger cortical area, in fact, is not true. We couldn't find evidence for that. And what we did find was that certain tracks, the better organized they are, as measured through FA, in the thalamocortical connections, do predict the how fast your alpha rhythm is. Much better than this that has no essential relation. You see here what is cortical surface and the peak of the alpha rhythm. It's just a random connection. Now, why is this important? Because the way to tie together multimodal data is through biophysical theory. That's what we think. Just let me give you an example from when I was a clinical neurophysiologist. When you zap someone to get the somatosensory evoked response to see if it was normal or not, you need to take into consideration how long his arm is. People with shorter arms, if they have more or less normal conduction velocity, of course will have shorter latencies. One of the things that we need to work more is to use all that biophysical theory, and I hailed the virtual brain and things like that, is to put together the different imaging modalities. Now, this is one example. Other examples are, for example, this, which is a voxel-wise connectome that we obtained. We plugged in equations, and we did detailed simulations of a subject's EEG and fMRI by predicting what would be the activation with changing thalamic input. I don't have time to explain it. This is just to say that when you have the individual data, you can use anatomical data to tie together different types of functional data. So multimodal data bases with EEG are important, but I want to turn now to something which is why I think it's important at a health and world level. And this is my last argument. And the problem is that there's something the economists forget about, which is the mental wealth of nations. And the problems we have are, and the criticism I have of most of the major brain projects, is that you have neurosciences over here, you have the global burden of disease. Just 4 days ago, Lancet published Global Burden of Disease 2013. I recommend you look at it. And a huge part of that is due to brain disorders. And it's not just in the developing world where we have scanners and MRIs, it's all over the place. Very poor countries, okay? And as a matter of fact, poor countries contribute more to the global burden of disease. Now, what do we need? We need a bridge between neuroscience and the global burden of disease. And That bridge would be neurotechnology, be it the diagnostic or therapeutic, with diagnosis and treatment. But the problem is that if you only produce instruments for the third level, the highest level, the specialized hospitals, you're not going to impact on the global burden of disease. You have to develop instruments for primary health care. And that's where the EEG comes in. So this is the amount of people, and millions of people, that have different brain disorders. There's a huge contradiction between that immense, let's say, quantity of people effective and the technologies which are very costly. Cuba's come up with a scheme which we call hierarchical active screening, and basically it's screening the populations before they're sick. Medicine should be not in the fancy hospitals, medicine should be where people are and before they're sick. And what we've tried to do is to tune our instruments and then have electrophysiology, the EEG in the middle and neuroimaging in the top, and these are the health levels. This has worked in other things. For example, we've developed this system to test hearing loss, and here are some statistics of what we've managed to achieve in terms of changing health indicators. That's the next slide. This was when we started in the 80s, this hearing screening program. Here you have IQ, and this is the number of months when you detect the kids. Now, you can change health indicators with these methods. In fact, in Ecuador, we've helped screen more than nearly 500,000 kids. So I'm talking about, and this is EEG, this is electrophysiology. So I'm talking that the bridge should be electrophysiology, and ignoring electrophysiology and modern brain projects is really, it's really a loss. We've used it in learning disabilities. These are the large studies with the quantity of kids we've looked at, which we followed up with we can predict seeing what core abilities, I'm not going to go into details, just to show you in which context we're using. We're looking at the morphometry, of course, but what can we do in all the kids? We can do electrophysiology and create these schemes of detection and feedback. So I go on to my last part of this, because this is a problem that we have very seriously. Cuba, due to its health system, is going to become the oldest population in the Americas. That's in 2020. We'll be older than the Canadians, we'll be older than the people in the US. We have a huge problem with cognitive aging and with dementia. And we've been gearing our works towards this slowly, but not fast enough. I have to be critical about that. And for example, we already did one study using the EEG about 15 years ago. Some of you may know that Francisco Lopera, and we're having a meeting in December to launch with the University of San Francisco, California, San Francisco, and with Trinity College, a global initiative, and he's going there. He discovered 5 families that had the presenilin 1 mutation. So in this family, a person who has that mutation will, with 100% penetrance, have Alzheimer when he has 45 years approximately, okay? So what we did was to see if the EEG could be a biomarker for this by taking 3 samples within the family, 1 that did not have the mutation, the controls, 2 that had the mutations, but 1 that had slight symptoms, and 1 that had already some symptoms, not full-blown Alzheimer. And the task here, the pattern recognition here problem, was the multivoxel pattern analysis, was to try to predict whether the person had the mutation or didn't. Now, I want to show you here what can be considered. That's our EEG system 15 years ago, okay? Now, look here. This is the area under the ROC curve. And here you see the spectra on the scalp. What you're going to see is that those that don't have the mutation, don't have X theta, as they progress towards, not progress. If they have the mutation, they have slightly more theta. If they progress towards symptoms, it's even more theta. This is very similar to work that's been carried out in Italy. Now, here you have the area under the ROC curves to see if you can find out if they have the mutation or not according to the two groups, the carriers without symptoms and those that have some symptoms. And while statistically significant, they're not impressive. But if you use the average brain and you do source localization, you find that the differences are mostly in the left temporal region. And this is cross-validated. We're not double-dipping the data or anything. In fact, I would say this is a very early example, because we did this about 10 years ago, of molecular imaging but using the EEG instead of PET. And what we see on the right is that the classification performance increases immensely. This has led us to the idea that maybe the EEG can be a marker for health in populations. And what we've done now is to try to look at those people that did not enter as normal controls into our brain mapping project, but had hypertension, because you know the effects this has. Very briefly, it was roughly the same proportion. It's really staggering how many people in the world and in Cuba. This has to do with diet, race, lack of exercise. Anyway, we grabbed all these guys, and what we asked them was to contribute to a study which was to essentially try to see if we could find biomarkers for brain damage in otherwise normal and controlled hypertensives. You know that when you have large vessel disease, it's pretty clear-cut. When you have small vessel diseases, you need technology, and the technology is available for the heart and for the kidney. But who's thinking of screening with MRIs? That's ridiculous. So what we did was a study with about 50 of these subjects in which they volunteered. We gave them the works, that is to say, looked at retina, Doppler, you imagine it, and also EEG and MRI. Not surprisingly, though they were under control, 78% of them had white matter lesions, had atrophy, had enlargement of the virtual Robin space. Now, what's interesting here is that many people say these are just incidental findings. However, we found a close correlation for Wechsler and mini-mental tests according to the severity of this, the lesion load that we found. Now, here we see the statistics. The only thing to summarize is that there were many more people that had white matter lesions and atrophy and all these things than had kidney or heart problems. So this has led us to discuss with our Ministry of Health that there has to be a change in the policy, because when you have a person that has one of the target organs damaged, you have to change the therapy. Now, what we did then was multivoxel pattern analysis, trying to predict the lesion load. And of course, once again, this is super bootstrapped, all the stuff to avoid false results. You can ask me later about it, but here's basically the results of looking at all the biomarkers we measured. And what did we want to predict? Lesion load in the MRI. And here you see that a discriminant equation with slow wave activity in alpha was pretty much predictive of what was happening in the MRI. This did not surprise us. And then you have other things that diminish. The retina was surprisingly disappointing. What does this leave us? And this is the example. Okay, this is other stuff we're doing to show you that we're not ignoring, let's say, high-powered stuff. We've developed a compound that can be used in rats to detect whether there's beta amyloid. But the point of this, you can ask me about that, is that we think that for cognitive aging, the problem doesn't have to be dealt with when you're older. It has to be dealt with when you're in middle life. What we propose is, once again, that for this problem, there be a hierarchical screening program that will allow us to attack these problems. The strategy of the Cuban Neuroscience Center with multimodal databases has been to apply this approach, and I haven't had time, for the hearing loss, where we've also used imaging to see neuroplasticity, neurodevelopmental disorders, learning, cognition, and it's an approach that we want to share and also learn from other people that are doing similar things. Now, two things that will make this much easier to use. You can look at this poster, I think it's on Tuesday, and it's improved head models. As I told you, we pioneered using the Montreal Neurological Institute's average brain. Now we have a better idea. We grab an EEG, we do source localization, and we do it on the lead fields or heads of the whole sample we have. We pick those that are closest to, that give a better statistical evidence, and we average those. This is called Bayesian model averaging. And we're getting very good results, much better than using an average brain. Why do I mention this? If I'm going to be true to what we think, that is that we have to have tools that are not the MRI or PET or anything to use in public health, then we have to develop techniques, and this will follow only by detailed statistical modeling. The second thing, which you all know is happening, but Cuba has jumped into with some pals from Hungary, is developing dry electrodes. So this is a device we've been testing in the past few months, which has one, here, this is a dry contact, and then two carbon fiber electrodes, and we're planning on using this to see if we can use it as a primary screening device. Remember, if you do hierarchical screening, you can have a lot of false positives. You want to avoid false negatives. But then you have to have economically valid methods to screen out the people. So I just want to end, because I find it really ironic. This is what Carl classifies as the years. He said that neuroimaging and brain mapping started out with the PET years, the fMRI years, and then he sort of predicted that the future would be the EEG years and MEG years. So I hope that the symposium today and this presentation convinces you that this should have a space in the Human Brain Mapping Organization, and that it's certainly something that's not expensive and should be added to most projects. Thank you. This is what I wanted to present.
Hi, really nice talk. Is the data from the Cuban study available for download, to download the data?
We haven't, okay, you know that Cuba's had a problem that fortunately is getting fixed with the internet. It's fortunately, I think, thanks to the reapproachment between Cuba and the US. But we're making it available, okay? But if you ask me, I can give you, it would just have to go on a disk, okay? I may be preaching to the converted already, because if you're here in this course, it's because you're interested in it. But anyway, transmit the message. It's really sad not to see EEG used in many different studies. Everywhere I go, I ask, and it's the simple translational tool you can use, okay? Okay, we can discuss later, okay?
I briefly want to wrap up the entire course now. Are there other questions that are left that you would like to address now? Can we get a very general feedback on the course overall? Did we meet those things that you were looking for? Did you get the information that you wanted? Who felt served by the course that we have done today? Who got roughly what you wanted to hear? And what are the things that we should add that we're missing from your point of view, that we just want to improve for the next years? Are there proposed? Yes, please.
So I initially hoped for more multimodality. I know it's not quite in the line of the talk we just heard, but more fMRI and potentially other modalities.
Okay.
So.
Yeah. Okay. Yes.
Actually, I would like to suggest for the future editions to include a part on realistic head models, because it was mentioned that that was important. When you do connectivity, you have to reconstruct activity in the source space, and in some cases you cannot do it in patients, for example. So I think that BAM models were used, FAM models were used, but yeah, there could have been a session dedicated to this.
A session, say it again, particularly on?
How to build realistic head models, what
Yeah.
is the advantage of one model compared to the other,
Okay.
and how you could deal with lesions in the brain, for example,
Okay.
how to estimate the conductivity of the different tissues.
Yeah. Okay. Yes. Other suggestions, other gaps in the course we did today? No. Okay. And thanks for the suggestions, and we will take this serious. And we close the course. Thanks for everybody for being here.